Okay, so welcome back to this next video in which we are discussing Alzheimer's disease and Alzheimer's disease medications. Okay, so what we have now discussed is some of the large-scale changes that are seen in the brains of people suffering from Alzheimer's disease. What we're now going to study is some of the much more small changes that are occurring. Okay, so we're going to see what you can actually see if you look at the cortex of the brain of someone suffering from Alzheimer's disease. Okay, so remember the cerebral cortex is where the cell bodies of the neurons are actually located. So there are two key findings, okay, two key features that you see, okay, and these were both described by Alzheimer in 1901. Okay, so the first is what are known as amyloid plaques. Okay, so let me explain what an amyloid plaque is. Now, importantly, these are extracellular, okay, so they are not inside the neurons themselves. Okay, so let's draw some neurons, okay, and then we'll draw some extracellular amyloid plaques. Okay, so we'll have a neuron here. Here is its dendrite. Here's its nucleus, well, its cell body rather. Okay, and here is another dendrite, and then we'll have its axon coming off here. Okay, like so. So this is a typical sort of neuron in the cerebral cortex here. They often have these very triangular shaped cell bodies here, and then there's their nucleus. Okay, so here is our neuron. Okay, now, in the brain of someone suffering from Alzheimer's disease, what you will find in the areas surrounding the neurons are extracellular amyloid plaques. Okay, so what is an amyloid plaque? Well, basically, it is a lump of proteinaceous material. Okay, so that's one of the first things. It's proteinaceous. It's a lump, and it's made up of protein, proteinaceous material. Okay, and basically it's loads of little fibrils, which basically just means a little fibre, okay, that have aggregated together. So here is a fibril, and we'll discuss how these fibrils are actually produced later on. For now, just understand that it's a little fibre, okay, a tiny little fibre, okay, known as a fibril. Okay, and what's going to happen is these fibrils are going to all line up, basically, okay, like so. And they're going to form a huge, great aggregate, basically. And this sort of a structure, which I'll highlight in orange here, this whole thing then is what's known as an amyloid plaque. Okay, so this is called an amyloid plaque. Now, some of them, like this one, will be located, far away from this neuron. But if they occur very close to the dendrites of a neuron, or even the axon of a neuron, what can happen is they can cause the dendrites or axons to swell. Okay, so let's say that this dendrite here is actually going to end up with an amyloid plaque surrounding it. Okay, what generally happens is you get a huge swelling of the dendrite where the amyloid plaque is surrounding it. So let's say this is the surrounding amyloid plaque here, and then this is the swelling of the dendrite. So to show this, I'll colour in the dendrite in green here. So the cell, this neuron is now being coloured in green. Okay, here it is, like so. Okay, well, outlined in green. And now we've got this swelling surrounding where the amyloid plaque is here, which I'll colour in orange, which remember is this aggregation of all of these fibrils together. Okay, right, so neurons don't seem to like these amyloid plaques at all. Okay, right, we will discuss the actual origin of these amyloid plaques later, but for now just note that they are a histological feature if you're looking at the cortex under the microscope of someone with Alzheimer's disease. Okay, right. Now, that's not the only feature. The other feature, which I'll have to now put in above amyloid plaques, but the amyloid plaques we think are more central to the pathology of Alzheimer's disease. Okay, we think this next one is a result of the amyloid plaques. Okay, so the next one is neurofibrillary tangles. Okay, and again we will discuss our theories as to where these things come from later on. Okay, so neurofibrillary tangles are abbreviated as NFTs. Now, what are these? Well, basically, these are intracellular. So the amyloid plaques were extracellular. The neurofibrillary tangles are going to be intracellular, although we'll discuss that they can end up being extracellular later on. Okay, so what are neurofibrillary tangles? Well, basically, they are these messes inside cells. Okay, so you can see cells that are just full of loads and loads of fibrils again, okay, or they're called a different thing this time, but it's the same principle. They're called filaments this time, okay, which are very thin little threads that are made up by proteins, which we'll come on to later. And this time what's going to happen is these little thin threads are going to aggregate into what are known as paired helical filaments. Okay, so here's one of these filaments, and it's going to form a sort of spring-like helix, okay, an alpha helix as it's called. That's the fancy word for a spring structure. Okay, and then what you're going to have is another one intertwined amongst it, okay, which will also be in a spring-like structure. Okay, so if I colour these two in, one of these filaments is in red here, okay, and the other one is in green here. Okay, and together this structure is known as a paired helical filament. Okay, and for short, paired helical filaments are called PHFs. Okay, so P for paired, H for helical, and F for filaments, so PHFs. So in neurofibrillary tangles, you get these filaments forming, and they assemble into these paired helical filaments, and you just end up with loads of these things, sort of forming a meshwork inside the cell body and also the dendrites of this axon here. So if I show them in vivid purple, so here they are. Here's a single paired helical filament. Here's another paired helical filament. Okay, you'll get them all over the place. Basically, you'll get these PHFs all over the place in the dendrites and just forming a huge, great neurofibrillary tangle. Okay, so when you get loads of them forming a great tangled mess inside the cell body and dendrites of a neuron, that's known as a neurofibrillary tangle. Okay, that's another feature of the brain of someone with Alzheimer's disease, a microscopic feature. Okay, and as I say, we will discuss where the neurofibrillary tangles and where the amyloid plaques actually come from later on. Okay, right. Now, neurofibrillary tangles start off intracellularly, but they can end up being extracellular. Okay, so you can end up with neurofibrillary tangles like so, extracellularly located. Now, how does this occur? Well, basically what happens is some of the neurons that have these neurofibrillary tangles, they then die. Okay, remember, it is fundamentally a neurodegenerative disease. This is a neuron that's still alive, but it's clinging on for bare life, basically. It's not doing well. Its prognosis is not good, and it's surrounded basically by neighbours that have had the same thing happen to them, and they've ended up dead, basically. And now their neurofibrillary tangles that were inside them are now just like a tombstone forevermore here, basically. Okay, so neurons which are clinging on for bare life in this degenerating area, which is full of these extracellular amyloid plaques, are full of neurofibrillary tangles, and they're surrounded by these neurofibrillary tangles which are now extracellular, which are basically the tombstones of other neurons which have now already degenerated. Okay, so that's the histological appearance then of an area of neuron cell bodies in the brain of someone with Alzheimer's disease. Okay, right. So we will come on to the neurodegenerative process that underlies Alzheimer's disease later. We will see our understanding of where these amyloid plaques come from and where the neurofibrillary tangles come from. Okay. For now, I'm going to take a step back though. What I'm going to do is I'm going to talk through where this degeneration is worst, okay? Which bits of the brain are affected most, okay? Because it is a whole brain disorder. You're getting neurons dying all over the place, okay? You're getting the weight of the brain going down, this brain atrophy, the cerebral ventricles are enlarging all over the place, okay? the gyri are getting smaller and the sulci are getting bigger. Okay, it's a whole brain disease. However, there are certain areas that are affected particularly badly very early on, and these are the portions which lose their ability to actually function first, and therefore they produce the symptoms of the early Alzheimer's disease. Okay, as of course the whole brain begins to degenerate more and more, you're going to lose more and more until eventually it kills you. Okay, but in the early stages, certain areas are affected first, and those produce the symptoms of the early disease. Okay, so I want to talk about that then first. Okay, so what we're going to start off with then, and indeed where the disease starts off, is it first affects areas areas that are going to be involved in memory. Okay, and the two areas that are particularly affected, which are involved in declarative memory, are the entorhinal cortex, okay, and also the hippocampus. So I want to describe the anatomy then of the entorhinal cortex and the hippocampus. Okay, now we're not going to go into our theories of memory in any detail. We're just going to describe the anatomy of these two structures and say, oh, it's involved in declarative memory. Okay, it's involved in memories that you can consciously recall. Okay, we're not going to go into the theories of how these areas actually work, because I would spend hours discussing how the theories of how these areas actually work. Okay, right. The hippocampus and entorhinal cortex are located in the medial temporal lobe. Okay, so let me show this on our familiar picture where we are viewing the left cerebral hemisphere from the left-hand side. Okay, so this is the temporal lobe here. Now the medial temporal lobe is not the side that we can actually see, it's the side that we can't see. In order to see the medial temporal lobe, we would have to be a little man standing on the temporal lobe here, and we'd have to walk round, okay, to the bottom of where we can see, and then we'd have to keep going, keep walking basically, and we'd eventually curve back round, and then we'd be on the medial temporal lobe side. It's the side that is facing inwards rather than outwards. We are seeing the lateral temporal lobe, the side that faces outwards. Okay, the side that faces inwards is the medial temporal lobe, and this is where the entorhinal cortex and the hippocampus are. Okay, so what I'm going to do then is I'm going to imagine chopping the brain down like so. Okay, I'm going to imagine taking a massive great carving knife and chopping the brain down like so. Okay, in a coronal section as it's known. Okay, so whenever you chop like so, where you're putting the front of your carving knife on one side of the brain and the back of your carving knife on the other side of the brain, that's known as a coronal section. Okay, then what I'm going to do is open the brain up and have a look at that new surface that I've cut open. Okay, and specifically I'm going to look at the temporal lobe here. Okay, so firstly then I'll draw out, starting with the parietal lobe here. Okay, so I'll show the lateral fissure. Okay, so this portion here that I've coloured in green, that represents that portion there. Then what's going to happen is the cerebral cortex is going to invaginate inwards in the lateral fissure here. Okay, and now we're coming down onto the temporal lobe cortex here in purple. So this is this portion here. And we're going to go completely round now. We're going to do what this little man was doing. We're going to go round, 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 and now we're on the bottom, and then somewhere down here you're going to have an important sulcus, which I'm going to mark out. Now I should say there are going to be other sulci along the way, which I haven't drawn, okay? I'm only going to draw the one that I care about here, which is the collateral sulcus here. Now I should just label up, this is the lateral fissure, this massive great crack or invagination of the cerebral cortex here. So this is the lateral fissure. Okay, then down here we have the collateral sulcus. Now, after the collateral sulcus is where we're going to start getting the beginnings of the entorhinal cortex, or the parahippocampal gyrus, as I'll explain in a moment, and then we're going to fold into the hippocampus. Okay, so this cerebral cortex here, okay, which I'm going to highlight in orange. This is going to be either the entorhinal cortex or what's known as the parahippocampal gyrus, depending on how far forward you are. Now, we're probably, annoyingly, just at the position where it would be about to interchange between the two. Maybe we would be in the entorhinal cortex region, okay? Basically, if you're far back over here, this portion of cerebral cortex, which isn't visible on this picture, I'm sorry, I've drawn it as though we're talking about the lateral temporal lobe here, okay, but we're not. We're talking about the medial temporal lobe. We're talking about this portion that isn't actually visible on this picture, okay, but imagine that this front bit of the temporal lobe is invisible and we're seeing this medial aspect of the temporal lobe. This portion of cortex corresponding to this portion here, okay, that would be the parahippocampal gyrus, okay, there. So if we were taking a section over here, we'd see the parahippocampal gyrus in this position here. So this would be the parahippocampal gyrus, okay, and I'm just going to go straight through that, I don't think it will harm me, okay, the parahippocampal gyrus. Okay, now if you go forward of the parahippocampal gyrus, further forward here, this portion, and it's just the continuation of the parahippocampal gyrus going forward, this then is known as the entorhinal cortex. Okay, so it really is just the continuation of the parahippocampal gyrus. So depending where we'd taken this cross-section, we would either be seeing the entorhinal cortex, or we'd be seeing the parahippocampal gyrus. Okay, so we're kind of almost in the switch point between them. So I've coloured it in the same colour as I've shown the parahippocampal gyrus, so we'll say that it's the parahippocampal gyrus. Okay, but if we were further forward, this would be the entorhinal cortex. Okay, right. Then it's going to continue curving back round like this. Okay, and then we're going to come into the hippocampus here. Okay, like so. It's going to curve right round like this, then you're going to have a swelling here. Okay, and you're also going to have another portion that wraps round like so. Now, not all of these structures that I've showed here are actually part of the hippocampus strictly, okay? They are part of what's known as the hippocampal formation, okay? But often when people say hippocampus, they mean hippocampal formation. I'll talk about that more later on once we've talked about all of these different structures here. So, after the parahippocampal gyrus or entorhinal cortex here, what you're going to have is an area known as the subiculum, okay, which is coloured in here in blue. Now, this is one of these areas that is not strictly speaking part of the hippocampus, but is part of the hippocampal formation. However, when people say hippocampus, they generally mean hippocampal formation, so they generally assume you realise that they mean the subiculum as well. Okay, so then after the subiculum, we're going to have the first area which is actually part of the proper hippocampus. Okay, here. And this area is known as CA1. Okay, now all portions of the hippocampus have the same nomenclature. They all have this CA. Okay, now what does CA stand for? Well, it stands for cornu ammonis. Okay, so this stands for cornu ammonis. Okay, now this in some old language, I would imagine Latin, okay, means ram's horn. Okay, and the reason that all the portions of the hippocampus are named cornu ammonis 1, cornu ammonis 2, cornu ammonis 3, cornu ammonis 4 is because it kind of looks like a ram's horn is what people thought. Okay, so ram's horn here. Okay, so this is the first area of the hippocampus. Okay, then after CA1, you're going to have CA2 here. Okay, so a second portion of the hippocampus. Okay, then we've got another portion of the hippocampus here after CA2, shown in orange. That's CA3. Okay, and then after CA3, this swelling here is CA4. Okay, so these four portions, CA1 to CA4, these are the hippocampus. Okay, so strictly speaking, when people say the hippocampus, that, strictly speaking, should be what they mean. Okay, generally that is not what they mean, however. Now, sometimes if people want to be, if they actually do want to mean the strict definition of hippocampus, some people will put hippocampus proper, okay, and then if someone says hippocampus proper, you can assume that they really do mean just CA1, CA2, CA3, and CA4, okay, because generally when people say hippocampus, they don't just mean those 4 bits, they mean CA1, CA2, CA3, CA4, along with the subiculum here in blue, and along with this banana-shaped thing that I haven't yet named. Okay, so let's now discuss this banana-shaped thing here. Okay, so this portion in green, this is the dentate gyrus, okay, and this is the final portion of what's known as the hippocampal formation. Okay, so this is the dentate gyrus. Okay, so now to do the strict nomenclature. So, hippocampal formation then. This is the strict nomenclature to mean the hippocampus proper, CA1, CA2, CA3, CA4, plus the subiculum and the dentate gyrus. So all of those portions there, that is what is strictly meant that's what should strictly be called the hippocampus formation, or the hippocampal formation rather, okay, rather than just the hippocampus. However, when people do say the hippocampus, generally they mean the hippocampal formation, so they mean the subiculum along with the CA1 to CA4 and then the dentate gyrus. Okay, right, so that's the anatomy then of the hippocampus, or the hippocampal formation if you like, because we will use hippocampus in this sense, i.e. meaning hippocampal formation, and the entorhinal cortex. Okay, so these 2 areas, which the anatomy of which we've just looked at, they are incredibly involved in declarative memory, okay, and other forms of memory as well, but especially declarative memory. Okay, so this is one of the areas that is particularly badly affected by Alzheimer's disease very quickly. Okay, so even in the early stages of the disease, when neurodegeneration has not been too bad everywhere else, the hippocampus and the entorhinal cortex, they're much further ahead basically. They are particularly susceptible, so they degenerate very quickly, and that's why in the early stages of Alzheimer's disease you get loss of declarative memory. Okay, right. We will call it there for this video, and in the next video what we will move on to is another area of the brain that is particularly affected by Alzheimer's disease early on, okay, and how the loss of this area is going to lead to problems with cognition.